This video follows up the scientific method video, going into a little bit more detail about experiment design and data presentation, that kind of thing. So we talked about control groups in the previous video. Let me just make sure that I'm good to go here. Yep. Control groups and the experimental group. These groups should be as identical as possible. The things that are the same between them are called the controls. Diet, exercise, etc. These are examples. You might be varying the temperature between things. You might be changing the diet because you might be interested in diet. You might be changing the exercise routine. But anything that you keep the same between these groups is going to be the control. So let's say for this experiment that I'm setting up, I'm keeping all of these things exactly the same between the two of them. Maybe even some other things, like if we're experimenting with rats, we'll use the same breed of rat. We will make sure that the male-female ratio of the rats is the same. We'll keep it 50-50, exactly the same. We'll make sure that the age of the rats is the same. Now, if you're doing this with humans, you can't get a genetically identical population, but what you can do is get a very, very large sample size. And hopefully, if it's a big enough sample size of randomly selected people, if you have some people that are very, very tall, and some people that are very, very small, and some people with a fast metabolism, some people with a slow metabolism, all of those tiny little fluctuations will average out. And if you have two big groups, they'll be relatively similar to each other. That's the best we can do. Variables are those things that are going to be different between those groups. So in my experiment, we're going to give them a medication. That's going to be the variable that's different. Now, what does that medication do? Let's say it affects their blood pressure. I'm interested in changing their blood pressure. So that means that I expect their blood pressure to be different, and that would make that a variable also. If at all possible, when you're designing an experiment, you only want one variable that you are controlling, and then one variable that you are measuring. There could be multiple variables that you're measuring, but you want to only be affecting one variable as the experimenter. In this case, the medication they're receiving. A different experiment might have the one variable be the exercise regimen, another one might be the temperature that, say, some bacteria are being incubated at. Whatever it is, you have one variable that you control, and then you can measure the response of other variables, things that change between the two groups. These have different names. So you have the one you control, and we call this the independent variable. This is the independent variable, the one that you, as the experimenter, have some degree of control over. It's the one that you have decided to influence. Blood pressure, though, you don't really directly control the blood pressure. Changes in blood pressure between these two groups is going to come about as a result of the medication that you give them. So this variable depends on the other variable. The dependent variable depends on the independent variable. So just to clarify this, the independent variable is the one that you are controlling to some degree. The dependent variable is the one that you are measuring. This one is measured. This one is, I want to write controlled, but since we're talking about control groups as well, that might be confusing, so I'm going to say manipulated. The independent variable is the one you manipulate. The dependent one is the one that responds to that manipulation, responds to that independent variable. There's also two different kinds of control. Well, there's multiple different kinds of controls, but there's also two more kinds of controls that I want to tell you about, and they are called positive controls and negative controls. I'm going to just bring us down a little bit. So let's say I have a mystery substance. I want to know what's inside this mystery substance. It's in a test tube. We're going to call it substance X. Now, what I can do in order to figure out the identity of substance X is I can add an indicator. Let's say I want to know if there's any sugar inside substance X. And the way this sugar indicator chemical works, I'm going to add a couple drops of this indicator chemical to substance X, and I'll get one of two results. Either I will get what we call a positive result, which in science, positive doesn't necessarily mean good, it just means that you confirmed what you were looking for. The thing you were looking for was present. And negative, which would mean that it's not present, it's not there. It doesn't mean it's bad, it just means it's not there. So how would I tell? Well, the indicator turns color in the presence of sugar. So positive, you would get a color change. Negative, no color change. But how do I know I can actually trust the results of this experiment? Because here's a secret about scientists. We don't trust anything. We don't believe that our tools are working properly. We don't believe that our experimental design is beyond reproach. We don't even believe our own logic is bulletproof and beyond reproach either. We assume that at every step along the way, somehow there will be errors creeping in, there will be bias creeping in, there will be mistakes being made, and that there will be accidents along the way. So how do we know that this experiment gave us the correct result? And one way that we double check is by using what are called positive and negative controls. First thing we want to do is double check that this indicator is working correctly. We want to make sure that if it's in the presence of sugar, it does change color, that it gets a positive result. In order to do that, we need what we call a positive control. And in this case, it's pretty easy to think up what the positive control would be. A substance that we could add the indicator to and definitely get a positive result. Can you think of a substance like that? Sugar. Yeah, it's going to be sugar. Maybe you said glucose or sucrose or fructose or something like that, but just any kind of sugar because I'm keeping it vague. So if we take sugar, which will be our positive control, we expect to see a color change. So, why do we do this? If the sugar indicator molecule changes color in the presence of sugar, we know that it works. We know that it actually does the thing that it purports to do. It does successfully change color in the presence of sugar. So positive controls are there to prevent false negatives. Let's say we spend days, weeks, testing different kinds of chemicals, and we find that none of them contain sugar. And then after all of those weeks, we find out that actually the indicator won't even change color if we add it to pure sugar. That though all of those negatives that we got, those were false negatives, that it wouldn't have worked even if there was sugar present. So we have to throw away all those results. So we use a positive control, to make sure that we get good results, that we don't get any false negatives. All right, so what's a negative control then? A negative control is the opposite. It's something that we expect to give us a negative result. So let's say I take another substance and add my sugar indicator to this substance. But I'm trying to come up with something that will definitely not get a color change, something that I know for a fact has no sugar inside of it. You could pick a number of substances for this purpose, but the standard one, the easiest one to go for, is going to be water. Just add it to pure. There's no sugar inside of water. Water is just hydrogen and oxygen. It's got no more complicated molecules in. Better yet, distilled water, which is really, really pure. All right, so we add that and we get no color change, which is, of course, a negative. So this is what we call a negative control. Now, let's be clear about this. A positive control is not every substance which gives you a positive result. It's something that you're using to test your indicator. It's something that you're using to make sure that the experiment works properly. So sugar is a good positive control because your indicator molecule is there to test for sugar. That's how you know that it's going to give you a positive result. Negative controls are not all substances which give you negative results. It is the one that you knew for a fact contained no sugar in this case. It did not contain the factor being tested, and so you knew it was supposed to give you a negative result. If you get a positive result with your negative control or a negative result with your positive control, there's something wrong with your experiment. So we use these in order to make sure the experiment's working properly. Just like positive controls prevent false negatives, negative controls prevent false positives. How would it do that? Well, let's say that every single molecule that we tested over a several week period of time, all of them were showing up as containing sugar. Just color change, color change, color change, color change. Then we did this test with just pure water and we still got a color change. Well, that would mean that there's something wrong with the test. It probably means that there was sugar inside the test tubes and they weren't properly cleaned out the first time. So we do this kind of test to make sure that our positive results are actually positive results. So that's the difference between a control group, controls, and your positive control and your negative control. Now, I also talked about dependent and independent variables. So let's talk a little bit more about them. So let's say we were talking about that study with the mice and the blood pressure medication, all that stuff. I finished the experiment, I got some results. How would I graph those results? How would I present them? I would start out by creating a shape kind of like this. Should look familiar. These are the two axes of your graph. There we are. And you guys probably know that this is called the x-axis. And this is called the y-axis. Now, let's say I had, results that look kind of like this. That looks like some results. It's going in a direction, certainly. And you intuitively know that this is probably the time axis, and that this is the blood pressure axis. Now, time and blood pressure are both variables. And remember, earlier in this video, I told you that you can have independent variables and dependent variables. Independent are the ones that you are in control of, that you manipulate. Dependent are the ones that you measure. We measured blood pressure. So that is the dependent variable. The dependent variable always goes on the y-axis. So time, did we manipulate time as our variable? We did actually. We were in control of how long the experiment went on for. So we were in control of how often blood pressure measurements were taken. We were in control of how long the trial went on. We were the ones in control of the timing of the experiment. So that means that time was an independent variable. The other way to think about an independent variable, if it's not something that you feel like you're directly manipulating, it's the variable that's not being affected by the other. You could say something like, what was the mouse's blood pressure? And someone could say, well, it depends. It depends on what? It depends at what time you are talking about. So the dependent variable depends on the independent variable. But you couldn't flip it around. You couldn't say, what time is it? And someone would be like, well, it depends. And you'd be like, depends on what? Depends on what blood pressure the mouse has. That's a nonsense statement, right? Especially because let's say that we had our control group, mice, right? And their blood pressure went like that. So here, be like, well, if the time depends on what blood pressure they are, they have the same blood pressure at several different time points. So was it multiple times at the same time? It doesn't make sense that way is the point I'm trying to get at. On this graph, I actually sneakily worked in a third variable, and that is group. Green was the control group, and the blue was the experimental group. Group is another independent variable. It's something we had control over. Even if we randomly assigned them, we decided how many groups there were going to be, what the nature of the groups were going to be, the different treatments that the two groups were going to get. We are in control of it. So let's take a look at another kind of graph. This is what we call a line graph. And a line graph is really good for showing change over time or variation over a temperature. It's really good when you have one variable, in our case time, that is changing consistently over the course of this graph, and then the other one that is responding to it as that changes. So you could have temperature and say growth patterns are going up and down, all that kind of stuff. But let's say I wanted to just look at what the average blood pressure was between the two groups by the end of the experiment and compare them. I would use a different kind of graph for that purpose. In that situation, I would have, again, we have the dependent on the y-axis, and then we have the independent on the x-axis. In our case, we just want to compare the final results. Our independent variable is going to be our groups: experimental group, control group. This is a bar chart. Really good for just getting a good idea of the comparison between two groups. You can see the difference between those final values. And they'll often have a little something on them. They'll have something that looks kind of like this. And these are error bars. And they will tell you how much error the scientists think is in that experiment or in those measurements there. If the error bars don't overlap, it's not something to absolutely go by, but it looks real nice. We could say that, right? If we could say that this number we're pretty confident is actually below this number, so long as those error bars don't overlap, because the error bar is indicating that the real value could be pretty much anywhere in between these two locations, So that's a nice thing to have. Now use a bar chart if your independent variable is qualitative. In this case, the experimental group and the control group, we are describing them. We are using adjectives to describe them. That's qualitative data. But one last one that I think is important for you guys to know about is called a scatter plot. In a scatter plot, my description of dependent and independent variables starts to fall down a little bit, because you're going to take two measured bits of data. So let's say that we were looking at people's SAT scores. That's a measurement. We tested them. We got a number back from them. We didn't know it was going to be ahead of time. Versus their income at age 50. And for each person, we check their SAT scores. If someone gets low SAT scores and they have low income at age 50, it's here. If they have high SAT scores, high income at age 50, it's up here. And kind of in the middle of the road would be somewhere like here. And for each person, we create a little data point. And what you'll start to notice is that, yeah, there might be a little bit of a trend, but it's not perfect. It's not a perfect trend. There are some people who had high SAT scores with low overall income at age 50. Some people who had low SAT scores and ended up with really high income at age 50. But if you take a look at the overall direction of this data, and I try to find a line that puts about half the data on one side and half the data on the other, I would say maybe it would look like something like this, like that. The question is, how tightly does the data group around that line? If it groups really, really tight, like there's a lot of data points right around that line, let's add some to create the impression that it's really, really tight, then that means that there's probably a pretty strong correlation between those two features, between SAT scores and income at 50. By the way, I just made those two things up. I have no idea if there's even any data on that topic. In general, I don't think SAT scores are a great predictor of your success. But this would be an example of really strong correlation. There's a lot of data points right next to that line, very few scattered away. But if it looked like just static, the whole page was filled up with little dots, that would mean that there's basically no correlation between those two things. This is what we call a scatter plot. And after line graphs, I would say that this is one of the most common graphs that you will see in science, is a scatter plot like this. Okay. Now, use a scatter plot when you have two measured values and you're trying to see if there's a connection between those measured values.